In Drosophila, the specification of the dorsal ventral axis or back belly axis, like that of the anterior posterior axis, also begins before fertilization. As with the specification of the anterior posterior axis, the specification of the dorsal ventral axis also involves the protein gherkin. And that protein is deposited into the oocyte from the nerve cells before fertilization. Remember that gherkin becomes localized initially to the posterior of the oocyte, and it signals to the posterior follicle cells, which respond by sending signals that reorganize the microtubules. And recall that the reorganization of the microtubules allows different factors to be apportioned to the anterior and posterior regions of the oocyte. As the cell expands, the nucleus moves from the posterior region to the dorsal anterior region, and it brings with it the gherkin message. The stainings on these embryos show gherkin, and as you can see, it's localized to the future dorsal-most anterior region of the embryo. The gherkin mRNA, which moved with the nucleus, eventually becomes translated into protein, and that gherkin protein binds to receptors on follicle cells that are surrounding the future dorsal region of the embryo. And recall that the follicle cells are part of the egg case. They're not part of the embryo. However, the gherkin signal influences the signaling pathways that are expressed in these cells and also the factors that they secrete. So the effect of this is that different signaling pathways are activated in the dorsal and ventral regions of the oocyte. And we're not going to discuss all the details of these signaling pathways, but the important thing to keep in mind is that the localization of the gherkin mRNA to the region of the embryo that's going to become the dorsal side is what results in the activation of different signaling pathways in the ventral side compared to the dorsal side. And the activation of these different pathways then leads to the specification of different fates along the dorsal ventral axis. In the embryo, the protein that distinguishes the back from the belly is the product of the gene dorsal. And remember that in Drosophila, genes are named after the phenotype that results when they're deleted. So when dorsal is absent, the embryo is dorsalized. So that means that it lacks ventral structures. If the embryo lacks ventral structures when dorsal is absent, that suggests that dorsal is needed for the specification of the ventral fate. This protein acts as a morphogen. Its activity is going to be highest at the ventral side of the embryo. But an important thing to keep in mind is that dorsal is actually found everywhere in the embryo, not just in the dorsal side. So how can it be acting as a morphogen? Throughout the embryo, dorsal is bound to another protein, cactus, which prevents it from entering the nucleus, so it's acting like an inhibitor. So cactus prevents dorsal from moving into nuclei and activating the transcription of genes that are needed to specify the ventral fate. On the future dorsal side of the embryo, where gherkin is present, the toll signaling pathway, which is needed to promote the degradation of cactus, is inactivated. So cactus remains bound to dorsal in the future dorsal side. So that means that the dorsal remains inactive. In the future ventral side, where the gherkin protein is absent, the toll signaling pathway is activated, and that's what you're seeing here. And this signaling pathway allows for the cactus to be degraded and dorsal then to become free to localize to the nucleus. In these ventral nuclei, dorsal initiates a transcription of genes that are going to be needed to specify the ventral fate. So in the wild-type embryo, dorsal will enter the ventralmost nuclei. If dorsal is deleted, the ventral structures fail to form and the embryo becomes dorsalized. On the other hand, if dorsal is allowed to enter every nucleus, then dorsal structures do not form and the embryo becomes ventralized instead. I mentioned previously that dorsal acts as a morphogen, and this is because the activation of dorsal occurs in a gradient, with the most ventral cells having the highest levels of dorsal localized to the nuclei in that region. The region that has the highest levels of activated dorsal becomes the mesoderm, and those cells where less dorsal has localized to the nuclei become the nerves and the glia. So in insects, the nervous system is located in the ventral side of the organism rather than in the dorsal side.